last week. We're still talking about membrane structure, membrane function, structure, and transport. And last week we already talked about the function of membranes, and then we also talked about both the composition of membranes. We said two major components being phospholipids and proteins. And then we talked about the structure of a membrane. And then we kind of started on membrane transport. So I introduced to you the different types of transports, also the different types of molecules that are able to pass through a phospholipid bilayer without any help versus those that are going to need help getting across the membrane. And then we ended by talking about two forms of transport. So let me go back to the list of different forms of transport. So you have passive versus active. And we said there's going to be two forms of passive transport that we talk about, simple diffusion and facilitated diffusion. And then there's going to be two forms of active transport that we talk about. That's going to be primary versus secondary. So the first two forms of transport that we're going to talk about are going to be simple diffusion and facilitated diffusion, which, given their name, both involve diffusion. So what I'm going to do is show you an animation that you saw in one of the pre-lectures that you did for this topic a while ago, and then we'll define diffusion for this class.
Molecules dissolved in a solution are in constant random motion due to their kinetic energy. One result of this motion is that dissolved molecules become evenly distributed throughout the solution. This tendency of molecules to spread out is an example of diffusion. But how do these molecules come to be evenly distributed? Let's start with a beaker of plain water. What will happen if we now add a lump of sugar to the water? A lump of sugar is composed of many individual sugar molecules, and even as a solid lump, the individual sugar molecules are in motion. When the lump is dropped into the water, it begins to dissolve. Individual sugar molecules move randomly and constantly from the area where they are common to the area where they are scarce. This type of motion, when molecules move from areas of their higher concentration to areas of their lower concentration, is called diffusion. Diffusion continues until all the sugar molecules become evenly dispersed throughout the beaker. The rate of diffusion is affected by temperature, size of molecules, and the steepness of the concentration gradient. Although not specifically shown in this animation, this is one of the processes whereby materials are exchanged between a cell and its environment.
Okay, so, yeah. Mm-hmm. If you just drink it, sorry, what?
Like, let's say that you pour the Kool-Aid powder in the water and you drink it while stirring it. Is it like how like the high concentration is still in that one particular spot? We know how like wood still tastes like, you know how like it still tastes like water in the way filled with sugar.
Well, I mean, if you're stirring it, that's going to move the molecules around itself. But then also there would be diffusion happening. So if you just dumped it and didn't stir it, eventually the molecules would diffuse throughout the solution evenly. But stirring it speeds up the mixing process. Okay, so they define diffusion for you in the animation. This is how we're going to define it for this class. So diffusion is the net movement of a substance from where its concentration is high to where its concentration is low. So in diffusion, the substance is moving down its concentration gradient. So remember, we talked about the concentration gradient last week simply being a difference in concentration between two areas. And if that something moves down a concentration gradient, it's going from where its concentration is high to where its concentration is low. So that's what's happening in diffusion. Something's going from where its concentration is high to where its concentration is low, so it's moving down its concentration gradient. Okay, they also, in the animation, mentioned diffusion rate. And I'll briefly define what we mean by diffusion rate. I'm not going to ask you to define it on a quiz or exam, but what we're talking about in diffusion rate is we're talking about how many molecules pass from one side of the membrane to the other side of the membrane in a given amount of time. Okay, so when you talk about diffusion rate across the membrane, you're talking about how many molecules are moving across that membrane in a given amount of time. And so they said there's three things, there's more than three, but three things they mentioned that affect diffusion rate. The first one was temperature. And this has the effect that you probably would expect it to have. So when you increase the temperature, the molecules are going to move around a lot faster in the solution, and that's going to increase the diffusion rate. So as temperature goes up, then I'm just going to abbreviate diffusion rate DR. Diffusion rate goes up. So as temperature increases, diffusion rate's going to go up, and then the opposite happens. If temperature decreases, then diffusion rate is going to go down. Okay, a second thing that they said affects the diffusion rate is the concentration gradient. So with respect to this, we're talking about how large is the difference in concentration between the two sides. Okay, and concentration gradient, like temperature, is directly proportional to diffusion rate. So as you increase the concentration gradient, that's going to increase the diffusion rate. So to say that in a different way, as you increase the difference in concentration between the two sides, the diffusion rate's going to go up. And then the opposite's going to happen. If you decrease the concentration gradient, then that's going to decrease the diffusion rate. Okay, so just as an example of that, let's say you have two scenarios where we're just dealing with a box that's divided into two areas. And on this side of the box, you have a 4 molar sodium chloride solution. And on this side, you have a 10 molar sodium chloride solution. And then in the second scenario, you have a second box also divided into two areas. And on this side, instead of having a 4 molar, you have a 2 molar sodium chloride solution versus a 10 molar sodium chloride solution, again, on this side. In which situation is the diffusion rate of sodium from the left side to the right side going to occur at the fastest rate? Right, the bottom one, because this has a larger concentration gradient compared to this, the concentration gradient that exists up here. So the difference in concentration between the two sides is larger here than it is here, and so the diffusion rate is going to be faster in the bottom scenario. And that's something that you guys actually observed in the lab that you guys did, I think it was 2 weeks ago now. You guys did the diffusion osmosis lab. And in one experiment, the first experiment you did in that lab, you detected more glucose in one of the solutions. And the reason for that was because there was a faster diffusion rate. And then also when you did the osmosis experiment, you saw that one of the bags gained a lot more weight than the other bags. And the reason that it gained a lot more weight again was because the diffusion rate was faster in that scenario. Okay, and then the last thing they mentioned in the animation that affects diffusion rate is molecular weight. They called it the size of the molecule. And this parameter is inversely proportional to diffusion rate. So as a molecule, with whatever you're talking about, if it's heavier, so as the molecular weight of the substance increases, the diffusion rate is going to go down. So what this means is that lighter molecules diffuse faster than heavier molecules. So lighter molecules are going to diffuse faster than heavier molecules. And conversely, as the molecular weight goes down, the diffusion rate is going to go up. Okay, do you guys have any questions about diffusion or diffusion rate? Okay. So this is kind of out of place. We already talked about this, but I'll just briefly go through it again. So this is something we talked about last week. So one thing that we talked about were concentration gradients, which is a difference in, sorry, it's not up there, a difference in concentration between two areas. And then we also talked about something called an electrochemical gradient. An electrochemical gradient is the combination of two gradients. So it's the combination of an electrical gradient, which is the membrane potential, and then a concentration gradient. So it's the combination of the membrane potential and the concentration gradient for a substance. And we said last week that the membrane potential simply refers to a separation of charge that can exist across the membrane. And I told you for a plasma membrane, it's typically characterized, the membrane potential is typically characterized by a slight excess of negative charges on the inside of the cell and a slight excess of positive charges on the outside of the cell. And so this is the electrical gradient. And this particular electrical gradient favors the movement of positive charges into the cell and negative charges out of the cell. Okay. And so when you're talking about the movement of charged particles or ions across the membrane, both their concentration gradient is going to affect their movement, but also if there's a membrane potential, that will also affect the movement of those molecules across the membrane. Okay, and then the next slide is just kind of a description of some of the functions for electrochemical gradients in biology. So they have a lot of important functions. The one that we touch on in this class is how an electrochemical gradient is going to be the power source for one form of active transport. So we said active transport requires energy, and one form of active transport that we're going to talk about gets its energy from an electrochemical gradient. Later on in the class, we'll also see how an electrochemical gradient is used as the power source to make ATP for the cell. So in both those cases, the electrochemical gradient we'll learn this actually today, is a form of potential energy, and you can use that potential energy to do something. In one case, you're using it to transport something against this concentration gradient, and in this case, you're using it to synthesize a molecule. One of the things that's interesting but that we won't touch on in this class is how electrochemical gradients are used in nerve signaling, specifically sodium and potassium electrochemical gradients. Neurons have the ability to produce electrical signals, and they do that by allowing both sodium and potassium to move across their plasma membrane that produces an electrical signal. And then also it's involved in muscle contraction too. Okay, so those are just some of the important functions of electrochemical gradients. So what we're going to do now is talk about our first form of transport, which is called simple diffusion. And as it says on the slide there, simple diffusion does not require energy, and it also does not require a membrane protein. Okay, so simple diffusion does not require energy and also does not require a membrane protein. And what happens in simple diffusion is you have a molecule that simply passes through the cell membrane from where its concentration is high to where its concentration is low. So that's all that happens in simple diffusion. It just simply passes through the membrane and from where its concentration is high to where its concentration is low. So in simple diffusion, again, something is moving down its concentration gradient. And then another important point is that the concentration gradient determines the direction of movement in simple diffusion. Okay, so again, the concentration gradient is going to determine the direction of movement. As an example of that, so two examples at the bottom of the slide of molecules that move by simple diffusion are oxygen and carbon dioxide. And what happens in your body is the oxygen that you inhale when you breathe is going to be transported by your blood throughout your tissues to the cells of your body, and oxygen is going to diffuse into the cells. And the reason that oxygen diffuses into the cells is because oxygen's concentration is higher on the outside and lower on the inside, so it continually diffuses into the cell down its concentration gradient by simple diffusion. And then carbon dioxide in your body actually moves in the opposite direction. There's a higher concentration of carbon dioxide on the inside of cells, so it's going to diffuse out of the cells down its concentration gradient. And then what happens is the carbon dioxide diffuses out of the cells and into the bloodstream. Your blood carries the carbon dioxide back to your lungs, and then you exhale that. Okay, so both of these molecules are moving by simple diffusion down their concentration gradients, but their concentration gradients are determining whether they move into the cell or out of the cell. Okay, so you should remember the three examples that I have down here for molecules that are going to move across a membrane by simple diffusion. So the two I just mentioned, oxygen and carbon dioxide, and then also fatty acids. And if you remember back to last week when we were talking about what types of molecules are able to get across the membrane without any assistance, that's going to be typically uncharged or nonpolar molecules. And oxygen, carbon dioxide, and fatty acids are all nonpolar molecules. And we said the reason that they can simply pass through a membrane is that they're highly lipid soluble. So they're highly soluble in this region and are able to just simply pass through a membrane. So those are the types of molecules that are going to be able to move across by simple diffusion because they do not need the aid of a protein to move them from one side of the other from one side of the membrane to the other. Okay, do you guys have any questions about simple diffusion? Okay, so before we get into the other types of transports, going back to the fact that there's some molecules, like I just mentioned, that can pass through a cell membrane without the aid of a protein, but then there's going to be other molecules that on their own are unable to pass. And those would be ones that are poorly lipid soluble. And those are going to be things like ions and then also polar molecules. And so you're going to have a protein that's embedded in the membrane in order to transport those types of molecules across the membrane. And we can just call those transport proteins. So these are proteins that are embedded in the membrane that transport a substance from one side of the membrane to the other. And we're going to talk about two types of transport proteins. One type is going to be called channels, and the other type is called carriers. And then you can call them transporters. So there's two names for the second type, carriers or transporters. These are just two types of transport proteins. So they both are going to move something across the membrane, and we will eventually talk about how they do that. Okay, so one thing that they have in common that you should realize is that each type of transport protein, either channels or carriers, are specific for what they transport, or said a different way, they show specificity for what they transport, which means that each of them is only going to transport either one or a couple types of solutes. So as an example, in your body, you have something called voltage-gated sodium channels. These are channels that, when they're open, only allow sodium ions to pass through them. They won't allow potassium ions, they won't allow chloride ions, or any other type of ions, only sodium ions when they're open. So they show specificity for sodium ions. And carriers are the same way, they show specificity. So you have specific carriers in the membrane of your cells that specifically transport glucose and only glucose, and not other types of monosaccharides and not amino acids. They're specific for that particular monosaccharide and only transport that one from one side of the membrane to the other. So they both show specificity for what they transport. They transport in slightly different ways. So channel proteins, I'm going to stay on the slide, when they're open, form a channel in the membrane, or you could say they form a pore. So what happens when a channel protein opens is it forms a pore in the membrane, and then whatever it allows to pass through it simply flows through the channel. So you can picture the way a channel moves something as allowing something to flow through it. But remember that it does show specificity. It doesn't allow anything to flow through it. It only allows a type of molecule or ion to flow through it. Carrier proteins act slightly differently. So with a carrier protein, you're going to have the carrier protein that has one conformation, and by conformation I mean the shape of the protein. And then what happens is the molecule that the carrier transports binds to the carrier, and that causes a change in shape of the carrier protein that allows the substance to be dropped off on the other side of the membrane. So you can see that it's in one shape here. After the molecule binds, it switches, and then the molecule or ion is passed to the other side of the membrane. So they act slightly differently from each other, but they both will get something from one side of the membrane to the other. Okay. Do you guys have any questions just in general about the two types of transport proteins? Okay, so what we're going to go into now is that there's actually different types of channels, which we'll go into first. And then there's different types of transporters that are present inside cells. So we're going to start with channels. So as it says at the top there are channels that always remain open, and those would be called leak channels. But then there's other channels that are going to open and close in response to a particular stimulus. And there's three examples. You have ligand-gated channels, voltage-gated channels, and mechanically gated channels. And they're all named after what they respond to. Okay, so those are all channels that open and close, and they're going to differ from each other in what causes them to open or close. So in a ligand-gated channel, what happens is these two channels here in the picture, the two blue channels, are ligand-gated channels. And ligand-gated channels respond to a molecule binding to the channel. So a separate molecule binds to the channel. So in this case, it's these two red molecules, and that causes the channel to open. And then the same thing is over here. These two molecules bind to the channel, causing it to open. And the reason they're called ligand-gated channels is because you call the molecule that binds to the channel a ligand. That's also what we're going to call a molecule that binds to a receptor is also called a ligand. So that's a ligand-gated channel. It responds to a molecule binding to the channel. Okay, so the second type of channel that you have are voltage-gated channels. And these types of channels respond to changes in the membrane potential. Okay, so these channels respond to changes in the membrane potential. So I previously told you that in your bodies, for the typical cell in your body, you have a slight excess of negative charges on the inside of the plasma membrane and a slight excess of positive charges on the outside. And you can actually measure in volts the membrane potential of the plasma membrane of a cell. And for a typical neuron, you don't have to remember this number, but for a typical neuron, when you measure it, it's minus 70 millivolts. And neurons, not all cells in your body, but neurons have the ability to change that membrane potential. And so what can happen is if the neuron, so this would be what's called its resting membrane potential at minus 70. And then what can happen is that it can be stimulated, for example, by another neuron, and then it changes its membrane potential to a minus 50 millivolts. And what happens in neurons is that when the neuron goes from a minus 70 to a minus 50, there's voltage-gated channels in the plasma membrane that open up and then allow a particular ion to pass through them. Okay, so this change from a minus 70 to a minus 50 causes the channel to open. And then what happens is eventually the neuron will go back down to minus 70, and as it's going back down to minus 70, the channels close back up in response to that change in membrane potential. Okay, so those are voltage-gated channels. Do you have any questions about those? Okay, so then the last type are mechanically gated channels. So these are going to respond to some type of mechanical stimulus. And two examples of mechanical stimuli would be pressure on the membrane or stretch of the membrane. So if you apply pressure to the membrane and that opens up a channel, that would be a mechanically gated channel. If you stretch the membrane and that opens up the channel, that would also be a mechanically gated channel. And you have mechanically gated channels in your ears that are involved in allowing you to detect sound. So that would be one example in your body where you could find mechanically gated channels. Okay, do you guys have any questions about the different types of channels? Okay, one other thing about channels before we move on to transporters that make sure you make a note of is that channels only allow for passive transport. Okay, so make sure you jot that down. Channels, no matter what type of channel you're talking about, it only allows for passive transport. So what that means is that if a channel opens up, it only allows something to move from where its concentration is high to where its concentration is low. So nothing ever goes from low to high through a channel. Okay, so channels only allow for passive transport. Okay, so now we're going to move on to carriers. And we'll talk about three types of carriers that are illustrated on the slide. So this is a picture taken from your textbook. The three types are on the slide here. So one type is called a uniporter. The second type is going to be called a symporter. And the third type is called an antiporter. They're pretty easy to remember. So a uniporter only transports one type of solute in one direction. So a uniporter transports one type of solute in one direction. So that's where the uni comes from, a single solute being transported. Both symporters and antiporters transport two types of solutes. So both symporters and antiporters are going to transport two types of solutes. Symporters transport the two types of solutes in the same direction. So you can see that the red circle and the blue triangle are both moving down in the same direction across the membrane. And so this transporter is called a symporter. And then an antiporter is a carrier that transports two types of solutes in opposite directions. So you can see that the purple circle and the red triangle are both being moved by this transporter, but they're being moved in opposite directions. One's being moved down and the other one's being moved up. Okay, and then what we're going to see as we go through this is that carriers, and this depends on the specific carrier that you're talking about, carriers are involved in both passive transport and active transport. Okay, so we're going to see that carriers can be involved in passive transport, but they're also involved in active transport as well. Okay, do you guys have any questions about the different types of carriers? Okay, so just one thing I want to mention before we move on is that this is not something that I would ask you on an exam or quiz, but it's something that I do want you to realize about a cell. A cell has the ability to change the permeability of its membrane. Okay, so what that means is the cell can change what it allows in and out of the cell. So one example of that that's pretty clear is that you have channels that can open or close. When the channels are closed, it's not going to allow that ion to pass through, but then when they open, now all of a sudden that ion can pass through, so the cell has changed its permeability to that ion. Another example of that happening specifically in your body is that when you eat a meal and your blood glucose levels go up, that ends up stimulating the release of insulin in your body, which is a hormone. And one of the things that insulin does is when it binds, so insulin is a signal, and when it binds to target cells, it causes those cells to insert a carrier in their membrane that transports glucose into the cell. So then when insulin goes up in your body in response to an increase in glucose, that causes the cells in your body to take in more glucose, and then that helps to lower your blood glucose levels back down. And that would be one of the things responsible for lowering your blood glucose levels back down after you've eaten a meal. So initially, after you eat the meal, it goes up, but then your body's going to return it back down to normal. So that would just be another example of cells changing their permeability and then allowing something that couldn't get into the cell before now to be able to get into the cell. Okay. So then the next form of transport that we're going to talk about, so the only one we've talked about so far is simple diffusion, and the second one is called facilitated diffusion. And the only difference between facilitated diffusion and simple diffusion is that facilitated diffusion requires a membrane protein. Okay, so when you say facilitated, diffusion has to be facilitated by something. And in this case, it's either facilitated by a channel or it's facilitated by a carrier, both of which are membrane proteins. So that's going to be the difference between facilitated and simple. Facilitated requires a membrane protein. But like simple diffusion, there's no energy required from the cell. So in facilitated diffusion, there's no energy required from the cell. And then in facilitated diffusion, you always have something moving down its concentration gradient. Okay, so something in facilitated diffusion, always moving down a concentration gradient without the input of energy. So what you have in facilitated diffusion is you have, so one example would be a molecule passing through this channel from where its concentration is high to where its concentration is low is facilitated diffusion. Diffusion is facilitated by this channel. This is also facilitated diffusion, where a molecule is moving from where its concentration is high to where its concentration is low, but you have a carrier that's involved in that transport process. So in this case, this carrier is facilitating diffusion in that case. So facilitated diffusion can be mediated by either channels or carriers. And then the last thing, yeah. No, that's close to what I said. Facilitated diffusion does not require an energy input from the cell. So the cell doesn't have to provide any energy for facilitated diffusion. Also, the cell doesn't have to provide any energy from simple diffusion. For simple diffusion and facilitated diffusion, the energy that's required for the movement comes from the concentration gradient of the substance that's being moved. So that concentration gradient we saw as potential energy, and that's used as the form of energy in this simple diffusion and facilitated diffusion, and the cell doesn't have to add anything to that. And then the last thing, two more things. One thing is that like simple diffusion, the concentration gradient is also going to determine the direction of movement, meaning does it go into the cell or out of the cell? That's going to be determined by the concentration gradient. On what side of the membrane is the concentration of the substance higher versus being lower. And then examples of molecules that move by facilitated diffusion, these are going to be molecules that require a membrane protein to get from one side of the membrane to the other, which means that on their own they're not able to simply pass through like the molecules in simple diffusion. So examples of that are ions and then also polar molecules. They can't simply pass through a membrane. They're going to require a membrane protein. So if they want to pass through a membrane by diffusion, it's going to have to be by facilitated diffusion. Okay, do you guys have any questions about facilitated diffusion? Yeah.
So you're saying that only facilitated diffusion, that it can only transport down?
Down a gradient, yep. Yeah. Facilitated diffusion is always going to be down a gradient from high to low concentration. And the same thing for simple. It's always down a concentration gradient. And so previously when I was describing the fact that when insulin's released, it causes those glucose transporters to be inserted in the plasma membrane of your cells, those glucose transporters perform facilitated diffusion. So they just let glucose enter the cell down its concentration gradient, and that's going to be facilitated by the carrier that gets inserted into the membrane. Okay. So the next type of membrane transport that we're going to talk about is active transport. And so this type of transport is also going to require a membrane protein, but in this form of transport, it's specifically mediated by a carrier. So active transport is specifically mediated by carriers only. You never have a channel involved in active transport. So active transport requires a membrane protein, and it's also going to require energy from the cell. So it requires the input of energy. And the reason that it requires the input of energy is because you're moving something from where its concentration is low to where its concentration is high. That's like moving something, that's like moving water up a waterfall. So that's going to require energy. And so in active transport, you have a membrane protein and an energy source within the cell that allows a carrier to move a substance from where its concentration is low to where its concentration is high. So the carrier moves the substance against its concentration gradient. And then examples of things that get moved by active transport include some of the things that also get transported by facilitated diffusion. Okay, do you guys have any questions about the general definition or what happens in active transport? Okay, so then there's two types of active transport. There's primary active transport and there's secondary active transport. And the difference between these two types, so just to back up a second, active transport is going to require an energy source. And then the difference between primary and secondary is what does the carrier directly use as the energy source. Okay, so the difference between the two is what is the direct energy source for the carrier. In primary active transport, it's simply the hydrolysis of ATP. So the carrier hydrolyzes ATP, and that releases energy that's used by the carrier to transport something against its concentration gradient. And then I do want you to remember a really important example in your bodies and then also the bodies of other mammals, and that's the sodium potassium ATPase. A lot of energy in your body goes to powering this transporter. Another name for this is the sodium-potassium pump. Sometimes you call carriers that perform active transport pumps. Okay, so sometimes carriers that perform active transport are called pumps. When you use the word pump when you're describing movement, that implies against the concentration gradient. So if you're on a quiz, or sorry, not on a quiz, but on an exam, and you're trying to define, let's say, facilitated diffusion, don't ever say that something's pumped in facilitated diffusion because you're telling me that's moving against its concentration gradient if you say pumped. So the sodium-potassium ATPase is a carrier that's present in the plasma membranes of your cells. And as the name implies, it transports both sodium and potassium, and it transports both of them against their concentration gradients. And for the cells in your body, typically, there's a higher concentration of sodium on the outside and a lower concentration of sodium on the inside. And then potassium is reversed. Potassium has a higher concentration on the inside and a lower concentration on the outside. And this transporter moves potassium into the cell, and it moves sodium out of the cell. So this is sodium, and then this is potassium. So the transporter moves both of those ions against their concentration gradient. So sodium is being moved out of the cell, potassium is being moved into the cell, and then this transporter or carrier uses the hydrolysis of ATP as its energy source for that movement. So it hydrolyzes ATP, and that provides the energy for that carrier to transport both sodium and potassium against their concentration gradients. Yep.
So sodium starts from a high concentration to a low, as you were explaining in the diagram.
No, they both have a high versus low, but for sodium, the high concentration of sodium is on the outside, and the low concentration of sodium is on the inside. But for potassium, the high concentration of potassium is on the inside, and the low is outside. Okay, just as one example of what this does, in your nervous system, so I told you that typically you have these concentration gradients for sodium and potassium across the plasma membrane of your cells. This pump maintains those concentration gradients. And those concentration gradients can be used by the cell for different things. As one example, neurons in your body use those concentration gradients to produce electrical signals. So if these concentration gradients were to ever dissipate, meaning the concentrations became equal on either side, then your neurons would not be able to conduct electrical signals and communicate with each other. So these would be really important. They're really important for most cells of your body, but but I just gave you a specific example of being important for neurons. Okay, do you guys have any questions about how primary active transport works or how the sodium-potassium ATPase works? Yep.
Because there's a high concentration of potassium on the inside,
Mm-hmm.
how is there an overall negative charge? Now you said there's a negative charge on the inside of the cell.
Yeah.
If there's a lot of potassium and it's positively charged, where does the negative charge?
All the proteins in the cell are negatively charged. But also this is positive too, and there's a higher concentration on the outside. A lot of the negative charge that's on the inside of the cell comes from negatively charged proteins. But then there's also negatively charged inorganic ions, but most of the contribution of the negative charge on the inside of the cell is from proteins that are there and other large molecules, not small inorganic ions like this. Okay, other questions? Okay, so then the other type of active transport is secondary active transport. And in secondary active transport, the carrier uses the electrical chemical gradient of one substance as the power source to move a second substance against its concentration gradient. Okay, so in secondary active transport, you have the electrical chemical gradient of one substance being the power source for the carrier to move a second substance against its concentration gradient. So what you're going to see happens in secondary active transport is the carrier transports two different substances. One substance is going to be moving down its electrochemical gradient, while the other substance is going to be moved against its concentration gradient. And as an example of, on the slide there, they gave you the example of a transporter that transports both glucose and sodium ions into the cell. And in this example, sodium ions are going down their electrochemical gradient because for the typical cell in your body, there's a higher concentration of sodium ions on the on the outside of the cell versus the inside. So sodium ions are flowing through this transporter down their electrochemical gradient, and that flow of sodium ions powers the carrier to also take glucose and move it against its concentration gradient. So this would be similar to the fact that you can use water going down a waterfall to power something. You're using sodium ions flowing down their electrochemical gradient as a power source to also move glucose against its concentration gradient. So if you have a cell in your body that has this specific transporter in their membrane, then another reason that the sodium-potassium ATPase is important is because this sodium-potassium ATPase maintains this concentration gradient for sodium ions. And if that concentration gradient ever dissipated for this cell, then this transporter couldn't work because it will have lost its energy source. Okay, so in this case, this is indirectly powering this transporter because this maintains the concentration gradient. This uses the concentration gradient as a power source. This happens in your kidneys. This is how glucose is taken up. So when your blood is filtered in your kidneys, a lot of what's filtered is then returned back to the blood by cells absorbing it. And this is one way that they absorb glucose. They absorb glucose using transporters that use the sodium-ion electrochemical gradient to move glucose into the cell also. Okay, do you guys have any questions about secondary active transport or the difference between the two?
So in the case for the secondary active transport, typically is it maintained by some primary active transport?
Yeah. Yeah. So typically, so the reason it's called secondary is because its primary source is the electrochemical gradient, but secondarily its source is ATP. So indirectly, this requires ATP, which maintains this, and then this is needed for that. So ultimately, ATP is also required for this movement. So that's why it's called secondary. Yeah. Okay. Other questions? Okay. This is just a picture of what I've already told you. It just gives you, you don't need to remember the numbers, but it gives you the specific concentration of 3 different types of ions for a typical neuron cell. But it just explains something I already told you. So, and that's showing you the sodium-potassium ATPase moving both sodium and potassium against their concentration gradient. But just to connect this with something that we talked about earlier, we talked about 3 types of carriers. So what type of carrier would the sodium-potassium ATPase be? Right, so the sodium-potassium ATPase would be a specific example of an antiporter. It moves 2 types of solutes, one solute being potassium, the other solute being sodium ions, and it moves them in opposite directions, so that would be an antiporter. Okay. So this just kind of summarizes what we've gone through so far. So we've talked about simple diffusion, where a molecule simply passes through the membrane without any help down its concentration gradient. Then we talked about facilitated diffusion, where something passes across the membrane down its concentration gradient, but it requires a membrane protein to do that. That could be either a channel or a transporter, which is also called a carrier. And then we talked about active transport, where something also requiring a membrane protein is moved against its concentration gradient from where its concentration is low to where its concentration is high. And then this form of transport requires the input of energy. Whereas the simple diffusion and facilitated diffusion do not require the input of energy. So that just kind of summarizes what we've talked about so far. Okay, so the next 2 types of transport that we're going to talk about, and we'll start by watching an animation about both of them, is endocytosis and exocytosis. And one thing that both of these have in common that separates them from previous forms of transport that we've talked about is both of these forms of transport are somehow going to involve vesicles. So remember, we defined a vesicle as simply being a small membrane-bound sac. So both of these forms of transport are going to somehow involve a small membrane-bound sac. So this will kind of animate that process for you.
The substances used as fuel by single-celled organisms include other smaller cells, particles of organic material, and large molecules that cannot pass through the plasma membrane. Many single-celled eukaryotes use a mechanism called endocytosis to ingest such food particles. In this process, the plasma membrane surrounds and engulfs the food particle. Cells use 3 basic types of endocytosis depending on the size and nature of the material to be ingested: phagocytosis, pinocytosis, and receptor-mediated endocytosis. If the material the cell takes in is particulate, such as a bacterial cell or an organic fragment, the process is called phagocytosis. If the material is a liquid, it is called pinocytosis. Some types of molecules, such as low-density lipoproteins, LDLs, are transported across the plasma membrane by receptor-mediated endocytosis. These molecules first bind to specific receptors embedded in the plasma membrane. The receptor molecules are concentrated in an indented pit coated by the protein clathrin. When sufficient target molecules accumulate in the coated pit, the pit deepens, seals, and is incorporated into the cell as a coated vesicle. Exocytosis is the reverse of endocytosis. This process results in the discharge of materials from membrane-bound packages that migrate to the inner surface of the plasma membrane, fuse with the membrane, and then release their contents to the outside of the cell.
Okay, so that described both endocytosis and exocytosis. It visually showed you how vesicles are going to be involved in both. So we're going to go, I'll describe endocytosis again, talk about the three types, and then talk about exocytosis. So just in general, no matter what type of endocytosis you're talking about, what happens is a portion of the plasma membrane is going to somehow surround something that's on the outside of the cell. So that's what you have happening in here, here, and here. This portion of the plasma membrane is going to surround these molecules. This portion of the plasma membrane is going to surround this bacterial cell. And then eventually what happens is that portion of the plasma membrane that's surrounding something on the outside of the cell is going to pinch off from the rest of the plasma membrane. And then that forms a vesicle that's now on the inside of the cell. And then the contents of that vesicle used to be on the outside of the cell. Okay, so in endocytosis, the product of endocytosis is a vesicle that's on the inside of the cell, and the membrane that's of the vesicle used to be a part of the plasma membrane. Okay, so then in the animation, they described three different types of endocytosis. There are more differences between the different types than we're going to go into, but for phagocytosis and pinocytosis, the difference that I want you to remember is what's actually being taken in, and they describe this in the animation. In phagocytosis, you're taking in something that's a particulate, like a whole cell or broken down fragments outside of the cell. And in pinocytosis, you're taking in a liquid. And then you obviously take in whatever is dissolved in that liquid. Okay, so sometimes phagocytosis is referred to as cell eating because you're taking in a particulate, whereas pinocytosis, because you're taking in a fluid, is referred to as cell drinking. Okay, so cell eating for phagocytosis and cell drinking for pinocytosis. Something you don't have to remember for an exam or quiz is that in terms of the cells in your body, all your cells are capable of pinocytosis. But you only have two cell types that are capable of phagocytosis, and that's macrophages and neutrophils, which are part of your immune system. And macrophages and neutrophils are both capable of doing this, taking in a bacterial cell. And what they do is that once they get the bacterial cell inside in a vesicle, they destroy the bacterium. So both of those cell types in your body are involved in clearing bacterial infections. Or getting rid of bacteria in your body. They do that through phagocytosis. And then the last type of endocytosis was referred to as receptor-mediated endocytosis. And as its name indicates, it's going to involve a receptor. And in this picture, it's the blue Y-shaped molecules there. And so the receptor is a protein that's embedded in the plasma membrane. And the extracellular part of the receptor recognizes a very specific molecule. So similar to the fact that channels and carriers show specificity, this portion of the receptor protein binds a very specific molecule on the outside of the cell. So what happens in this form of endocytosis is the small molecule that's recognized by the receptor binds to the receptor, and then that's going to cause that portion of the plasma membrane to take in that substance into a membrane-bound vesicle. And so the advantage of this form of endocytosis is this is a way that a cell could take in something specific that it wanted on the outside of the cell. So it would have these receptors in the membrane to recognize a specific molecule that it wants. Once the molecule binds to the receptor, then the cell takes it inside. Okay, do you guys have any questions about the different types of endocytosis? Okay, so then sorry, one more thing if it's not clear. So in endocytosis, I'll get you in a second, you're taking something into the cell. So it is important to remember that in endocytosis, you're taking something into the cell. Yeah?
Alright, so for the first two types, they don't need a receptor to like take whatever they want to inside the membrane.
Right. Okay. So exocytosis is a process that involves vesicles where you're going to release something from the cell. So you're going to, something's going from inside the cell to the outside of the cell. So with exocytosis, what you have is you start with a vesicle that's on the inside of the cell that contains a substance. So you start with a vesicle on the inside of the cell that contains a substance, and then the membrane of the vesicle is going to fuse with the plasma membrane, and it fuses in a way that dumps the contents of the vesicle to the outside of the cell. After it's fused, the membrane of this vesicle is actually a part of the plasma membrane then. And then the contents of the vesicle get dumped to the outside. One example, you won't have to give me examples of where this takes place in your body or in other cases, but one example in your bodies is your neurons release neurotransmitter by exocytosis. So neurons have this portion of their cell that's called the axon terminal, and you have all these vesicles sitting in the terminal that contain neurotransmitter. And then when that neuron is stimulated, all those vesicles fuse with the plasma membrane and release the neurotransmitter, and then that neurotransmitter binds to another cell, and then that's how it communicates to the cell that it's talking to. So that's one example of where it's taking place, where exocytosis takes place. Just an example of endocytosis where it takes place, I gave you the specific example of neutrophils and macrophages taking in a bacterial cell. But also we talked about the fact that there are certain single-celled organisms that use food vacuoles to eat. And that's going to happen through endocytosis. So the cell takes something into a membrane-bound vesicle that you then end up calling a food vacuole. And then that food vacuole fuses with a lysosome. And then that digests the contents of the vesicle so that the cell can use it. So endocytosis can be a way that single-celled organisms actually eat. These are other just examples of when endocytosis and exocytosis are used both in animals and then in certain cases plants. So you don't need to be able to list any of these for me for an exam or quiz, but just useful to know of when these things actually take place. Okay, do you guys have any questions about endocytosis or exocytosis? Okay, so then the last form of transport that we need to talk about is osmosis. This is something that you were introduced to in the lab. You did a whole experiment on osmosis. And osmosis is defined as the net diffusion of water across a selectively permeable membrane. So osmosis is the net diffusion of water. So it's when in osmosis, we're specifically talking about transporting water across the membrane. And in osmosis, in osmosis, it's going to be the net diffusion of water across a selectively permeable membrane. If you just want to remember the net diffusion of water, that's fine. Okay, now in osmosis, water is always going to, so it's the net diffusion of water. Water is always going to diffuse towards the higher solute concentration. So in osmosis, water is always going to diffuse towards the higher solute concentration. So if I gave you a scenario where you have a box that's divided by a selectively permeable membrane, and let's say you have a 4 molar sodium chloride solution on this side and a 10 molar sodium chloride solution on this side, and you make that membrane that separates the two sides impermeable to sodium and chloride ions, but permeable to water, in which direction is water going to diffuse in this scenario? Right, towards the higher concentration to the right. Okay, so I don't want to say 4, you guys know 4 is lower than 10, but you're going to diffuse towards the higher solute concentration. So that's one way you can remember it. The other way that you can remember it is that even in osmosis, something is going down its concentration gradient, and in this example it's water. So the higher the solute concentration, the lower the water concentration in a solution. So you have actually a higher water concentration here and a lower water concentration here. So water is in fact moving down its own concentration gradient. You just don't typically think of the concentration of water, so that's why I presented it to you as diffusing towards the higher solute concentration. But water is in fact moving down its own concentration gradient in osmosis. Okay, the next thing we're going to talk about is tonicity. This is also something that you were introduced to in the lab. And with respect to solutions and tonicity, you can have 3 types of solutions. You can have a hypotonic solution. You can have a hypertonic solution. And you can have, an isotonic solution. And in biology, when you're talking about the tonicity of different solutions, whether they're hypotonic, hypertonic, or isotonic, you're always comparing the tonicity of the solution relative to the tonicity on the inside of the cell. Okay, so you're comparing the tonicity of a solution relative to the tonicity on the inside of the cell. A more useful way to think about hyper and hypo and iso is thinking of these prefixes as referring to the concentration of solute in the solution relative to the concentration of solute on the inside of the cell. So again, a useful way of thinking about hypo, hyper, and iso in this case is that these prefixes apply to the concentration of solute in the solution relative to the concentration of solute on the inside of the cell. So in a hypotonic solution, the solution has a lower concentration of solute compared to the concentration on the inside of the cell. In a hypertonic solution, the concentration of solute is higher in the solution, hyper, relative to the inside of the cell, and iso means the same. So in an isotonic solution, the concentration of solute on the outside of this in the solution is the same as the concentration of solute inside the cell. Okay, and then all you have to do is combine that then with the fact that water is always going to diffuse towards the higher solute concentration in order to determine what type of effects these types of solutions are going to have on a cell. So we're going to see that a hypotonic solution, we said that the solution has a lower concentration compared to the inside of the cell. So when you put a cell in this solution, the higher concentration of solute is on the inside of the cell, so water diffuses into the cell towards the higher solute concentration. So a hypotonic solution is simply any solution that causes water to diffuse into the cell and the cell to swell. Okay, so a hypotonic solution is any solution that causes water to diffuse into the cell and the cell to swell. And again, the reason that that happens in a hypotonic solution is because water is diffusing towards the higher solute concentration, which is on the inside of the cell. And what you saw in the lab is that when you put red blood cells into a hypotonic solution that was just water, the cells swelled so much that they burst open and lysed the cells. Okay. A hypertonic solution, when you put a cell in a hypertonic solution, now the higher solute concentration is on the outside of the cell. So now water on the inside of the cell diffuses out towards the higher solute concentration, and then that causes the cell to shrink up. So a hypertonic solution is any solution that causes water to diffuse out of the cell and the cell to shrink. Okay, so a hypertonic solution is any solution that causes water to diffuse out of the cell and the cell to shrink. And that's what you saw in the lab with the red blood cells. When you put them in the hypertonic solution, the cells shrank up, and they did that because water diffused out of it. Yep.
So for the hypertonic, you're saying that is a low concentration inside the cell, right?
What?
The concentration is a low.
I didn't hear what solution you referred to.
Hyper.
For hypertonic, yes. No, for hypertonic, the lower concentration of solute is on the inside of the cell. Right. Okay, and then an isotonic solution, that's going to be any solution that causes no net movement of water, and then there's going to be no change in the cell. So an isotonic solution is any solution that causes no net movement of water. So when if you've ever been to the hospital and had like a saline drip, the saline that they give you in the hospital is isotonic to the cells of your body because you wouldn't want it to be hypo or hyper compared to your body. Okay. So this just describes the effects that you saw in the lab last week. In terms of putting a particular cell in a hypo versus hypertonic solution, so for red blood cells, when you put it in a hypertonic solution, water leaves and the whole cell shrinks. When you take the red blood cells and put them into a hypotonic solution, the cells are going to swell, and then if the solution is hypotonic enough, it can cause the red blood cells to also burst because of the water that moves in. Then you also did the same thing for a plant cell. But what happened with a plant cell is that when you put it into a hypertonic solution, all the water inside the cell, including the water that's stored in the central vacuole, diffuses out of the cell, and then the central vacuole shrinks up. And remember, the central vacuole is pushing everything against the cell wall. So basically everything moves away from the cell wall, so the plasma membrane tears away from the cell wall, and then the contents of the cell are in the center of the cell. So the cell wall stays in place, but everything else shrinks up. And then when you put a plant cell into a hypotonic solution, pretty much nothing happens because the cell wall will prevent the cell from expanding to a point where it's going to explode. Basically, it will prevent it from expanding at all. So a plant cell is not going to explode when you put it into a hypotonic solution. Okay. Okay, so the last thing I just want to discuss with respect to osmosis and tonicity is the fact, this is an organism that you guys actually looked at in the third lab, which is a paramecium. And I previously described to you the fact that protists, many protists, including paramecium, have a membrane-bound organelle called a contractile vacuole. We said that the purpose of that contractile vacuole is to get rid of excess water from the cell. And for these types of organisms, paramecium, they can live in a water environment that's hypotonic. So this is a single-celled organism, and they are capable of living in hypotonic environments. And the reason that they're able to do that is because they have that contractile vacuole. Because what happens is, if this paramecium is in a hypotonic environment, then water will be constantly diffusing into the organism. But the organism doesn't swell or explode because what happens is that water collects in a contractile vacuole, and then that contractile vacuole will get rid of that water that's contained within it, and that prevents the cell from exploding in a hypotonic environment. Okay, do you guys have any questions about the osmosis, tonicity? No? Okay, so let's do some of the lecture questions, and then we're only going to take a 5-minute break today because we're not doing a full lecture. But we're going to do the lecture questions for this topic first, and then we'll take a break. So the first few questions actually have to apply to things that were covered on the quiz today, but I do want to go through them again. So number 8, so we talked about the fluidity of the membrane. So just to review, what 2 aspects of phospholipids affects the fluidity of a membrane?
The amount of carbon double bonds.
Right.
The length of the.
Right. So the number of double covalent bonds between carbon atoms, and with respect to that parameter, the more double bonds you have, the more fluid the membrane is. And then the second aspect of phospholipids that affect membrane fluidity is the length of the tail. And with respect to that parameter, the shorter the tails, the more fluid the membrane. So 10 is asking a question similar to the one that you had on the exam, but it's asking the opposite in terms of the temperature change. So the temperature surrounding a cell increased. Which of the following changes could help to keep the fluidity of the plasma membrane constant? Right, B. So in this case, increasing the length of the fatty acid tails would help to keep the fluidity of the membrane constant, because increasing the length would decrease the fluidity and counteract the increase in fluidity caused by the increase in temperature. Okay, number 11. How would someone in the class describe the effect that cholesterol has on the fluidity of the membrane?
It's a buffer.
Right, and can you describe how you think of it as a buffer?
So basically, like as the temperature would increase, the cholesterol would decrease in order to keep, well, basically keeping a solution at a fluidity like the same.
Yeah. So you're right. At high temperatures, cholesterol decreases fluidity, and at low temperatures, it has the opposite effect. It increases fluidity. So what are 2 key differences? Number 17. What are 2 key differences between passive transport and active transport?
Active requires energy.
Right.
Carriers only. It only uses the carrier proteins.
Okay. So that is right. So active transport requires energy, and active transport only requires carriers. But there was another difference I was looking for when I asked that question. So I was looking for the requires energy.
The direction, like the active transport only comes from the high, like transport high from low to high.
Switch it.
High to low.
No, switch it. You said high to low first, so switch it. Low to high, yeah.
Okay. And then I think passive is from high to low.
Right. So those were the 2 key differences that I was looking for, was the energy and the direction of movement. So active transport requires energy, passive transport does not. And remember, what we're talking about is energy from the cell in that case. And then the direction of movement in passive transport, you're going down a concentration gradient, whereas in active transport, you're going against the concentration gradient. But you're right that active transport specifically requires carriers. Passive transport, it depends on the form. So simple diffusion requires no membrane protein. And then facilitated diffusion can be facilitated by either channels or carriers. So can someone describe to me for 18 what happens in diffusion? Or just give me the definition of diffusion is basically what I'm looking for. Yep.
It's the net movement of molecules from higher concentrations to lower.
Right. And what determines the direction that something moves when it moves by diffusion?
Concentration gradient.
Right, the concentration gradient. And then what are 3 things that affect diffusion rate that we talked about? Right. Molecular weight. So temperature, molecular weight, and concentration gradient, right? And remember, temperature and concentration gradient are both directly proportional. So as both of those things go up, the diffusion rate goes up. And if either of those things go down, the diffusion rate goes down. And then molecular weight is inversely proportional. So as the molecular weight of a substance goes up, the diffusion rate goes down. Okay, do you guys have any, are you guys okay with that?
Yeah, I'm lost on that, the third one, the weight part.
Yeah.
And so you're saying, so if there's, so what is saying that if some goes down, that means that the diffusion goes up?
No. So if you're comparing two different molecules and one molecule is heavier or larger than the other one, the smaller one diffuses faster than the heavier one. So the heavier one would have a higher molecular weight and diffuse slower. The smaller one would have a lower molecular weight and diffuse faster. Okay, so number 26, in what direction are things moving in an active transport again? Against what? Concentration gradient, right? So you're going from low to high. In what direction does a substance move in facilitated diffusion? Right, high to low, so down a concentration gradient. Okay, number 33, which of the following statements is true of membrane phospholipids? Right, they freely move laterally within the membrane. So the hydrophilic head groups, remember, face the exterior portion. The hydrophobic tails are pointed towards each other in the interior, so they're in the interior part. We said that phospholipids do not flip-flop very easily because in order to make that movement, you'd have to bring the hydrophilic head of the phospholipid into contact with the hydrophobic core. And then what would be the proper way, rather than nonpolar, how would you describe phospholipids? Right, amphipathic, so they have both a hydrophilic part and a hydrophobic part. Okay, so number 34, indicate if the following statements about transmembrane proteins are true or false. So A, they can aid in the transportation of molecules across membranes. True. Can you give me two examples? Two specific examples? No? So we talked about two types of transport proteins. Right, carriers and channels. So those are the two transmembrane proteins that are specifically do that job. Can they act as cell surface receptors? Right, yes they can, so true. This one you kind of have to think about it. It's not something that I told you directly. Do you think they are totally water soluble?
No.
What part of them do you think is not water soluble?
The part in contact with the extracellular environment?
Nope. Exact opposite of that.
The exact opposite?
Yeah. So so what did I ask? Did I ask what parts are water soluble?
Yeah.
Oh, then you're right. Sorry. Sorry, I messed that up. So the parts that are water soluble are the two parts that face either the fluid on both sides of the membrane. The part that's not water soluble is the part that's in contact with the hydrophobic core. So the part of it that spans the phospholipid bilayer is hydrophobic. So I didn't tell you this directly, but transmembrane proteins are also amphipathic molecules. So they have two hydrophilic parts and then a single hydrophobic part. And then can they be enzymes? Yes. They are proteins, yeah. But not all proteins are enzymes. And not all transmembrane proteins are enzymes, but some of them are. So this goes back, the cell surface receptors, we gave different functions for transmembrane proteins, which included being receptors, acting as channels or carriers, and then also we said they could function as enzymes. Okay, so number 36 is referring to different types of carriers. So what type of carrier binds two or more ions or molecules and transports them in opposite directions? Right, C, antiporters. And then for 41, you're supposed to determine whether the statement applies to facilitated diffusion, active transport, or both. So which one does not require energy? Right, facilitated. And which one do molecules move against their concentration gradient? Right, active transport. Which one requires a membrane protein? Right, both. Which one requires energy? Right, that's active. And then which type do molecules move down their concentration gradient? That's facilitated diffusion, right. Okay, and I think that's the, oh no, never mind, I have more. Number 43, which of the following statements is correct about active transport? It's not A. It's B. So molecules move from lower concentration to higher concentration. I didn't tell you D, so it does not require a gradient to exist across the membrane. It is not a form of diffusion because diffusion moves down a concentration gradient. C is false because it does require proteins, specifically carriers. And A is also false because it's not something that's going to happen spontaneously. It requires the input of energy. Okay, so number 44, sodium concentrations are higher outside and glucose concentrations are higher inside the cell. Using the same membrane protein for sodium and glucose transport, what is it called when a sodium ion electrochemical gradient is used to drive glucose transport into the cell against this concentration gradient? Right, B, that would be secondary active transport. Okay, so those are the last two. Do you guys have any other questions that you see in this list of questions that you want me to go through? Okay, so we'll take a 5-minute break, and then we're going to start with energy, enzymes, and metabolism. Mm-hmm. Yeah. Yeah.
Okay, so in my notes, I wrote that desmosomes connect cells to the extracellular adherence, and heavy
No.
desmosomes connect to the...
No. Yeah, that's wrong. So just let me double check. Desmosomes connect two cells together via, I just want to make sure it's cadherins. So yeah, desmosomes connect cells to each other via, so it's two cells for desmosomes.
So it does not connect to the extracellular...
Does not connect to the extracellular matrix. So whenever it's two, so in an anchoring junction, so for both desmosomes and adherens junctions, when two cells are connected together, it's always cadherins.
So what differentiates adherence junctions from desmosomes? They both connect to...
Oh, so one difference that we went to in class is how they're anchored on the inside of the cell. So what are they attached to on the inside of the cell? For adherence junctions, the junction is connected to actin filaments, but in desmosomes, they're connected to intermediate filaments on the inside of the cell. Mm-hmm. Yeah. So we have an incubator, and so the three, you guys do in 3 different temperatures, right?
Yeah.
Hot, cold, and room temperature.
25 degrees and-
Yeah, it's going to be 37 because that's what we have.
Yeah.
We have an incubator, a walk-in incubator that's 37 degrees, and I'll put those in there for you.
Yeah.
And then you're going to put obviously the 4 degrees you're going to put in the cold room, which is that room I normally go in to get solutions out of. That's at 4 degrees. And then the room temperature obviously just leave out on the cart that I bring out.
Okay, awesome.
Mhm.
Thank you.
Yep.